first video on DNA structure and replication. This is going to be a series of several videos covering all the topics related to DNA. So we'll talk about structure and replication, and then the following videos will talk about more of the functions of DNA, so protein synthesis and all of the topics that are related to that. So I thank you for watching, and feel free to leave a comment. So before we get started, there are a few things that you should know. Hopefully you've read in your textbook chapter 16 in the Campbell Biology, or you might have read chapter 14 in the OpenStax Biology book. And hopefully you've completed a reading guide, and then you're ready to just go over everything, make sure you understand what's happening. So some things you need to know about this section. First of all, if you have not read the history of the DNA race, if you've not learned about the scientists who sort of put the pieces together, then you really need to do that. It's an interesting story. So starting with T.H. Morgan, Griffith, Avery, Hershey and Chase, Chargaff, Wilkins, Franklin, and even Linus Pauling would be a good person to learn about, even though he's not directly involved. He's sort of a side story, and it's kind of a funny one. And then, of course, Watson and Crick. And after them, Meselson and Stahl, dealing with semi-conservative replication. And of course, you should know the structure of DNA. You should know what a nucleotide is. You should understand the difference between a nucleotide and a nucleoside, especially if you're in an advanced class, just because that's going to be helpful to you. So we'll go over that. And then also, you should know basic terms like pyrimidine and purine. You should know the nitrogen bases. You should know the sugar phosphate backbone, stuff like that. You should also know how many hydrogen bonds are found between the AT pairs and GC pairs, and you should also know what a phosphodiester linkage is. And lastly, we will cover DNA replication in detail. So let's start with a basic review of the parts of the DNA molecule. Just in case you need the review, you know that DNA is found within the nucleus, and that cells that don't have a nucleus still have DNA because they have their DNA organized into a region, usually called the nucleoid region. And we're just talking about bacteria. When we talk about cells that don't have a nucleus, those are prokaryotes. So in a eukaryotic cell, DNA is found in the nucleus. All right, so in case you don't know the words monomer and polymer, those are really important words you should already know, but a monomer is a single subunit of a larger molecule. So DNA and RNA are both polymers because they are composed of many small subunits. So this drawing I have right here is kind of a terrible drawing of a nucleotide. This is the basic monomer of a nucleic acid, and the two nucleic acids are DNA and RNA. And since this is a video about DNA replication, we're really just going to talk about the structure of DNA. So we're going to start off with naming the three parts that you should know. The first one is the phosphate. So I'm just going to draw a little P. That's really bad, sorry. This is a sugar, and since we're talking about DNA, this is deoxyribonucleic acid, so this is deoxyribose. It's a good thing I can spell. So that's deoxyribose, that's our sugar. And then over here, this rectangle represents our nitrogen bases. So I'm just going to write an NB. And the nitrogen bases, there are actually several different kinds of nitrogen bases, and in DNA, you've probably heard of this before. They're adenine, thymine, guanine, cytosine. Those are the four possibilities that could fit into this box here. And the box is a little bit misleading because it's really not shaped like a box at all. It's actually either a double ring structure or a single ring structure, and if you're an organic chemist, please forgive this terrible representation of the actual molecules. I'm not going to draw all of the little functional groups and things hanging out the side, but that's sort of outside the scope of what you need to know for an advanced biology class. So the double rings are a class of molecules called purines, and the purines include adenine and guanine. And then the single rings are pyrimidines. And the pyrimidines include thymine and cytosine. And then, of course, if we were talking about RNA, which we're not, we would talk about uracil, which is the third pyrimidine. Now, if you're from Texas like I am, you might notice some important colleges here. We've got the Aggies and we have TCU. If you want to remember that Aggies are pure, that should make somebody laugh out there. You can. TCU, all of these have a Y in their name except for you. Thymine and cytosine have a Y, and so does pyrimidine. So that's kind of how I remember it. And then the Aggies are pure. That's funny. I didn't go to either one of those schools, so I can't really claim any sort of inheritance there. Another way to remember this is that girls wear 2 wedding rings, and girls are pure, and guys just wear 1 ring, and guys have a Y, like a guy has a Y, or if you're a biologist, the XY chromosome. So there's that. Anyway, those are the purines and the pyrimidines. We really don't need that on there, do we? There it goes. Bye. These are, again, going to replace this box here. So in the overall structure of the DNA molecule, you're going to have many repeated subunits. So you're going to see this nucleotide over and over and over and over again. Now, a nucleotide has these 3 components, but a nucleoside is something a little bit different. It's just simply the sugar and the nitrogen base by itself with nothing. Now, a really famous nucleoside you might have heard of is a nucleoside triphosphate called ATP. It's kind of important. It's very, very, very important. It actually has 3 phosphates. That's what ATP stands for, adenosine triphosphate. So this would be an A. And then there are 3 phosphates. Well, interesting fact. What if I want a different nucleoside triphosphate, or NTP for short? Can I put a G here? I sure could. Then I have GTP, which is a really important molecule. Chapter 11 of the Campbell Biology book, Cell Signaling. If you're a 9th grader watching this, it's okay if you don't know what GTP is, but you'll probably learn about it at some point. You can substitute that for a T and have TTP, or you can have CTP, and then you could have UTP if this were RNA, but we're not talking about RNA. So interestingly, there have been some discoveries of some even other strange nucleoside triphosphates and nucleosides in general. That's the topic for another day. So interesting to note that DNA can actually provide its own energy when it assembles because it just uses nucleoside triphosphates. So you don't need separate ATP, you just need these NTPs, nucleoside triphosphates, to make ATP, which I think is super awesome. All right, so there's our basic monomer review, and now we've got what's going to become a polymer. Right now I have 4 nucleotides on the screen, and we need to connect them so that we can make a polymer. We're going to start off by adding the most important super strong covalent bond called a phosphodiester linkage. I'm going to add it right there, and I made it a different color because I want you to see that it's not the same as another bond we're going to talk about in a second. So there's another one. You see how they're running in opposite directions. That's another important fact. I'm just going to type in here the phosphodiester linkage. There we go. Just in case you want to spell that, because I think spelling is good. I know you can Google it, but you should know how to spell some things. So that's a really strong covalent bond. We do not want this backbone here to fall apart. We could add more nucleotides on the end here. We could just keep adding them. We can't add any to the top. We're going to talk about that in a minute. The other important place that we're going to hook these 2 chains together is between the nitrogen bases here. So remember, oh, I didn't want that to be the same color. We got to change the color. Can't have the same. Here we go. So that's really crooked. All my OCD friends out there are going to go crazy. Oh, there it is. I'm going to put 2 purple lines there. And I'm going to put 3 purple lines right here. And those of you out there, oh, see, look, they keep piling on top of each other. Those of you out there who are going, I know what that is, that's great. I'm really glad. These are hydrogen bonds, right? And the only reason that these are not crooked and these are is because I'm too lazy to fix it. Sorry, OCD friends. These are hydrogen bonds, and hydrogen bonds are different than covalent bonds. I'm just going to write H bonds. They are different than covalent bonds because they are weak and they are relatively easily broken and reattached. And as you might be aware, we have to be able to copy this molecule at some point so that we can make a copy of all of our DNA and then do mitosis or meiosis. So we've got to be able to break that and then put it back together. And so what is here in the middle? Well, we've got our, maybe this is an adenine. And maybe this one is, what does it have to be? Hopefully you know. It has to be a thymine because A goes with T, always together. If we want to have a GC pair, then we would just put them down here. Good combination. Now you may notice that the AT pair is here with the 2 hydrogen bonds and the GC pair is here with the 3. That's because there are 3 hydrogen bonds between G and C, but only 2 between A and T. So if I wanted to copy and make another nucleotide show up down here, I could, and then I could make it T, and then I would have A on the right. We would just keep doing that over and over again. The DNA molecule is ridiculously huge. A couple more really important pieces here that you need to know about before we move on to the next page. You do need to know that this end has a name. This is the 5' end, and this end is the 3' end. And since the 2 molecules run in opposite directions, that means this is a 5' end and this is a 3' end. And I always like to know why things are the way they are, so I'm going to let you know, not a little secret, it's not really a secret, but not everybody talks about this. I think it's important. So you know that this is a pentagon shape. It has 5 sides. These are numbered. So the reason that this is the 5' end is because this phosphate is attached to the 5th carbon. And surprisingly, the 5th carbon is actually sticking out over here. But that is the 5th carbon. Okay, 1, 2, 3, 4, and then 5 over here. And then the 3rd carbon is here. And so the reason this is called the 3' end is because it's right there by the 3rd carbon. And there's actually a little hydroxyl group hanging off here. And up here, the 5th carbon. So why is this important? The hydroxyl group is where we're going to add a new nucleotide. So if I want to add another one, I would just bring in my awesome drawing skills. Here it is. Oh man. What am I going to do here? I'm going to do a really important process that you've heard of before. I'm going to create my phosphodiester linkage, and when I do, I'm going to remove a water molecule. That is called dehydration synthesis. I'm hoping that you have heard that before and that this is a review. But if not, surprise, that's something you definitely need to know about. And it's really important because it's the way that all of our large biological molecules are synthesized. So we've got carbohydrates, lipids, proteins, and nucleic acids, the 4 major classes. And this process is common to all 4. So really should know about that. The opposite process, hydrolysis, I would just put the water back and then remove the bond, although it takes a little energy to do things like this, to put things together and break them up. So it's not quite that simple, but it is important. Enzymes are very helpful for stuff like that. All right, so this is how we build a polymer. If I want to add a new nucleotide on the right-hand side, it would have to go here on the 3' end, and then we would just keep building. All right, so that's the general idea of the structure of DNA. Next, we'll talk about the function of DNA replication.